Okay, now we will see the cell membrane. It is quite different from other bacterial and eukaryotic cell membrane, especially lipids. Normally, in other cells of bacteria, you will find that fatty acids are connected by ester linkage, while in this, it is a branched chain of hydrocarbon attached with glycerol. Archaeal lipids are derivatives of isoprenyl glycerol instead of glycerol fatty acid esters. It usually contains diether side chains of C20 carbons, means 20 carbons in length, and some are of tetraether chain of 40 carbons. Polar lipids are also present in archaeal membrane, cell membrane, like phospholipids, sulfolipids, glycolipids. From 7 to 30% of membrane lipids are also non-polar. This lipids can be combined in various ways to yield rigidity, to yield a membrane of different rigidity and thickness. For example, C20 diether can be used to make a regular bilayer membrane, while C40 tetraether lipids are used for monolayer lipids. Archaeal membranes may contain diether, tetraether, and other lipids, as might be expected for their need for stability. The membranes of extreme thermophiles, such as thermophilic bacteria, such as uh, thermoplasma, plasm, and sulfolobus, are mostly tetraether of monolayers. This is a diagrammic representation of three different categories of archaeal lipids. Here you can see that is ether bond instead of ester bonds and glycerol. It contains a glycoprotein structure, phytanols. They are also not phytanol glycerol diether. This one is diether structure, and this is our tetraether diphytanol chain reactions. uh, Chromosomes and ribosomes. The archaeal chromosomes have been studied uh, in a form of single closed DNA circle. The genome of archaea are significantly smaller than normal bacterium. E. coli Chromosomes and ribosomes. The archaea chromosomes have been studied as a single closed DNA circular. The genome of some archaeons are significantly smaller than the normal bacterium. E. coli DNA has a size of about 2.5 to 10 raised to 9 daltons, whereas thermoplasma acidophilus bacteria DNA size have been 0.8 to 10 raised to 9 daltons, while Methanobacterium thermoautotropicum have 1.1 to 10 raised to 9 daltons, and so it will be have a variations. Archaeal mRNA appears similar to bacteria rather than eukaryotic mRNA. It means messenger RNA. There is no evidence of mRNA splicing. Archaeal promoters are similar to those in bacteria. RNA splicing is a in a molecular biology is a form of RNA processing in which a newly made precursor messenger RNA pre-mRNA transcribed in is transformed into a mature mRNA. During splicing introns, non-coding regions are removed and extrons means coding regions are joined. This phenomena is normally present in eukaryotes, not in bacteria. Archaeal tRNA lacks thymine, contains pseudouridine or 1-methyl pseudouridine. The archaeal initiator tRNA carries methionine, as does the eukaryotic initiator tRNA. Although the archaeal ribosomes are 70s similar to bacterial ribosomes, electron microscopy studies shows that their shape is quite variable and sometimes differ from both archaea and bacteria. So overall, whatever the chromosome structures and the way of translating the messenger from one cell to its daughter cell is quite different. Like some archaea, such as many methanogens in the phylum Eukaryota, differ from the prokaryotes in having histone protein that binds to DNA to form nucleosomes like structure. There are large and complex enzymes which are insensitive to drugs such as rifampin and streptomycin. Thus, they distinguish from both the cells. Now, this is a quick summary of what is the similar between archaea and prokaryotes, and what are the similarity of archaea and archaea and eukaryotes. Like in prokaryotes, size, shape, and lack of nucleus, single chromosome, genus in operon, no introns. This all characters are quite similar to prokaryotes, while they are resembling in eukaryotes with having few plasmids, RNA, polymerase or promoters, then translation machineries, ribosomes and tRNA. Sequencing of Methanococcus jenischii in 1992, 56% of genus are not similar to bacteria or eukarya. Now we will see silent features of its group methanogens, halophiles, and thermoacidophilans.